Hello students, good morning. Let us see the muscle contraction. For the sake of contraction of a muscle, the muscle has to receive stimulus. Let us suppose this is a striped muscle fiber, and this striped muscle fiber is supplied with a nerve fiber, the junction of which is referred as neuromuscular junction, otherwise neuromuscular motor plate or motor end plate. Motor end plate. So, whenever a stimulus is given, a mechanical, chemical, thermal or electrical, because of the stimulus at the neuromotor junction, a neurotransmitter is released. Neurotransmitter is released. So, generally the neurotransmitter is acetylcholine. Acetylcholine is the neurotransmitter that is released at the junction of the nerve fiber and muscle fiber. Because of the acetylcholine, the permeability of the sarcolemma towards sodium ions is increased. As a result, the sodium ions present in the ECF are pumped inside into the muscle fiber. As a result, the calcium ions are released from the sarcoplasmic reticulum. Sarcoplasmic reticulum stores the calcium ions. Because of pumping of the sodium ions, the calcium ions are released. Whenever the sodium ions are pumped inside, an action potential is generated. An action potential is generated because of the depolarization. On the outer surface of the sarcolemma, depolarization is developed due to pumping of the sodium ions. As a result, calcium ions are released from sarcoplasmic reticulum. These calcium ions pass through the triad system, Triad system is formed by a T tubule and two cisternae present on either side of the T tubule. So, this triad system is responsible for the transportation of the calcium ions. When these calcium ions reach and attach to the troponin. So, we know that troponin is associated with the actin. Now, this is the actin protein, globular actin. So, this is G-actin, globular actin, and such two rows of globular actins together form actin filament. Now, it is having an active site. This active site is enclosed or covered by means of tropomyosin. This is tropomyosin. And tropomyosin is attached to another protein, regulatory protein, that is troponin. The troponin is differentiated into TNT, TNI and TNC. Calcium binding subunit of troponin, inhibitory subunit of troponin, and tropomyosin binding subunit of troponin. So, this regulatory protein is troponin. We know that the proteins of muscle contraction are actin and myosin, whereas the tropomyosin and troponin are regulatory proteins. So, these are the actin proteins which together form a filament. So, these are the two rows of the globular actins which form the filament, and this another filament like structure, that is nothing but the tropomyosin, and attached to the tropomyosin there you can see the presence of the troponin with the three subunits TNT, TNI and TNC. When the calcium ions are released from the sarcoplasmic reticulum, these calcium ions bind to the TNC subunit. When the calcium ions bind to the TNC subunit, the TNT pulls the tropomyosin aside. Thereby, the myosin binding site of actin is exposed. So, there is no tropomyosin here, and as a result, the head of the myosin fits into the active binding site or myosin binding site of actin to form actomyosin complex. It occurs only during muscle contraction. During relaxed state, the active site is masked by the tropomyosin, but only during contracted state the tropomyosin is pulled aside, so that active site is exposed. Immediately the head of the myosin fits into the active site of the actin. So, here in this diagram the mechanism of muscle contraction is being exhibited. So, the muscle contraction was explained by Hugh Huxley and Hansen. So, whenever the myosin binding site is exposed because of the calcium ions and the head of the myosin fits, we know that the head of the myosin is having two sides, one to bind with the ATP molecule and another to bind with the actin protein. So, because of the hydrolysis of the ATP, so here hydrolysis of the ATP is responsible for generation of high energy phosphate and ADP, and whenever it is exposed, whenever the active site is exposed, because of the energy it fits into the active site of the actin, and because of that energy the head of the myosin is pulled inwards in the active site. As a result, the actin molecules slide inwards because a large number of the cross bridges which are formed by the head and the neck or short arm are present around the myosin. So, the two actin filaments which are present on either side will be pulled inwards or slide inwards. For example, this is the myosin, and the myosin is provided with the myosin proteins. Each myosin protein is having the tail and also the head and cross bridge. They are directed in the direction of the jet membranes, to which the actin filaments are attached like this. So, these are the actin filaments. So, because of the cross bridges which fit into the active sites, and also because of the energy that is formed due to breakdown or hydrolysis of ATP, the actin molecules slide inwards around the myosin. Myosin is constant, only the actin molecules slide inwards along with the jet membranes. Even the jet membranes also move inwards. The myosin is constant, only the actin filaments slide inwards. So, because of the hydrolysis of the ATP, the energy is responsible for pulling the actin molecules inwards. And as a result, the ADP and high energy phosphate are released from one side, and from the other side one ATP molecule binds. When ATP molecule binds to the head, it is detached, and then the calcium is in a position to unmask the trypomyosin or pulling the tropomyosin to unmask the G actin, active site of the G actin, and once again it fits into the active site of the active site of the actin, and this cycle, cross bridge formation cycle, is repeated continuously, resulting in the muscle contraction. So, during the process of muscle contraction, the cross bridge moves to and fro. So, initially it is attached to the myosin binding site of actin. Because of hydrolysis, it is pulled inwards like this, and whenever the ATP binds to the head of the myosin, it is detached. Again it moves to the original position, and it will be attached to the another actin molecule due to hydrolysis of the ATP. So, this cycle is continued, and this head moves to and fro. It is otherwise referred as ratchet mechanism. Ratchet mechanism. The to and fro movements of the heads of the myosin proteins are referred as ratchet mechanism, that is responsible for the contraction of the muscle. So, this is the diagram showing the contraction and the formation of the cross bridge cycle. So, it will be repeated continuously, and as a result, so suppose this is the myosin molecule, and around the myosin, so these are the actin proteins, and these two are the Z membranes, and because of the combined action of all the cross bridges and due to the energy that is released due to the hydrolysis of ATP, the actin molecules slide inwards, thereby reducing the H zone. So, this is the H zone, that H zone gets reduced, and even the I band which is present on either side of the myosin, it also gets reduced because of the movement of the Z membranes inwards, and thus there is no differentiation of the I band and A band in the contracted state of a muscle. Here which is showing the muscle contraction. So, this is in the maximum contracted state. So, this is one sarcomere. So, here the sarcomere which is present between two Z membranes. So, this is Z membrane and this is Z membrane, having the myosin myofilaments and thin actin filaments, and this is the A band, and the middle there is H zone on either side of the A band, and these two parts constitute I band. And whenever the actin molecules slide inwards along with the Z membrane, so here there is no difference between the A band and I band, because the actin filaments slide inwards around the myosin along with the Z membranes. So, there is no differentiation of the H zone or the I band in the contracted state of a muscle. Now, the combined action of these cross bridges are ratchet mechanism, because these cross bridges move to and fro for about 50 to 100 times per second, as a result of which all the actin filaments that are present around the myosin, because we know that a single myosin myofilament, that is a single myosin myofilament. The single myosin myofilament is surrounded by means of 6 actin filaments in the form of hexagonal structure, and this is the myosin which is having the cross bridges. So, around each myosin myofilament we can say there are 6 rows of cross bridges. Cross bridges are formed by the heads, and of course, the head and the neck together form the cross bridge. And they are nothing but the components of the myosin protein. 2 types of proteins are there within the myosin, heavy muromycin and light muromycin. Light muromycin forms the main body of the myosin filament, and these are the heavy muromycin. So, they are nothing but the cross bridges. So, by the combined action of all these cross bridges, which exhibit this ratchet mechanism, or move to and fro for about 50 to 100 times per second, the actin molecules slide inwards. So, these actin molecules slide inwards, and as a result the muscle contraction occurs. And during muscle relaxation, the calcium ions re-enter into the sarcoplasmic reticulum. When there are no calcium ions, the troponin is not in a position to hold the tropomycin. As a result, the tropomycin masks the active site of actin, and the cross bridges no more can bind with the active sites. And as a result, the actin filaments move to the original position. This is relaxation. So, this is relaxed state of the muscle, and this is the maximal contracted state of the muscle. So, whatever that has occurred during contraction, during muscle relaxation, all those reactions occur reversibly. When there are no calcium ions, definitely the sodium ions will move outwards, and of course, there is no stimulus. When there is no stimulus and the sarcolemma is not in a position for pumping the sodium ions inside, and those sodium ions, when they pump outside, the calcium ions retain and re-enter into the sarcoplasmic reticulum. When there are no calcium ions, the troponin is not in a position to hold the tropomycin. Thereby, the myosin binding site of actin will not be exposed, and as a result, the heads of the myosin or these cross bridges cannot fit, and thereby the actin filaments move to the original position, and this is the relaxed condition. And that completes about the muscle contraction and relaxation. Thank you.